Good day. In this lecture, we will be talking about some basics of the docking mechanisms, particularly the basic molecular docking procedures. Okay, so first off, what is molecular docking? So molecular docking, as you can see here, the images here actually are the docking of spacecrafts in the space station. It's actually something similar like that. You dock something to a larger or even two objects that are of similar sizes. You dock them and you look at their interactions. Basically, when it comes to molecular docking is the study of how two or more molecular structures, usually it's a drug, a smaller molecule, the drug, we call this one the ligand, and the receptor, which is a protein, it can be also an enzymes, and we look at or how or whether if they fit together. So in molecular modeling, this is basically a technique that is used to predict. So most of these docking procedures are done as a predictive tool. So we want to know whether will they or won't they dock. So it's usually about the interaction of a larger molecule, which is our protein. Usually it's the receptor, it's the enzyme, it's the target of our study. And with a much smaller molecule, we call them the ligand. That ligand could be the drug that is used or applied. Okay, so somewhat similar like this one. So you have here a larger object. This could be your receptor, this is your protein, and then this could be your ligand. So we are looking at this tool, this technique is one for predicting, or at least we want to visualize whether or how they interact with one another. Now, why do we do this? So we have several reasons to do this. It could be both predictive or it can be analytical. So for predictive one, usually, or rather the most common application is in drug discovery and drug development. So when we say drug discovery, we want to discover new drugs that can be used for treatment of several diseases and conditions. So instead of doing large-scale screening of actual molecules in the wet lab, because these are very expensive. When you do screening, it usually involves lots of different types of compounds. Instead of synthesizing them or extracting them individually and purifying them individually in the lab, which is very laborious and very expensive, we just need to know what is the structure of our drug or the drug candidates. When we say candidates or screening, it involves hundreds, if not thousands, of possible drug candidates. And we look at them using docking, we can have an idea which of them can bind to our target molecule. This is our receptor, which has affinity to that receptor. Another one is in the development stage. Once we have found a candidate, or if there really is already an existing candidate, we can do optimization. Once we know what are the molecules, what type of interaction there are, we can optimize them so that either the interaction can be stronger, or if it's too strong, we can weaken the interaction. Why do we need to weaken the interaction? Sometimes a drug, um, because sometimes a drug is usually a poison. Let's say, for example, a toxin. A toxin that targets a certain ion channel that prevents the activation of an action potential. That toxin is very strongly binds to that particular channel. But we want to develop a drug, somewhat, let's say an anesthetic. We want to prevent, as an anesthetic, it prevents action potentials. But we do not want our drug to be very toxic, because that is originally a toxin, which is a toxic compound. It can kill you. But we want to develop a drug from that toxin. So we will weaken the interaction so that when we apply it to our molecule, when we apply it to that patient, the patient will not die because the interaction will be weakened. So instead of blocking the channel indefinitely, we just weaken it so that it can bind to the channel. It can block the channel for just a small period of time. So we can optimize basically that's optimization of this development. Okay, so another application could be a prediction of biological activity. We have a certain molecule extract from, for example, any medicinal plant. We want to predict what is that for, what can we use that for. So this is one of our reverse screening. So instead of different we have many ligands to one receptor, we have one ligand to many receptors. Other example could be we know that these two molecules interact. Are this interaction inhibitory or agonistic? When we say agonistic activity, the receptor or the molecule is activated when that ligand binds to it. When it is antagonistic, it produces a different action. Or antagonistic is it produces a different effect to the agonist. So agonist initiates an action, the antagonist inhibits the action. Okay, so it can also be used for binding site identification. For example, we know that this drug is a cure for, let's say, for example, this toxin is a blocks acetylcholine receptors. But we do not know where exactly. Why binding site identification? Because the receptor is very large. You will see later it's a very, very large compared to your drug. So in a very large landscape, we do not know exactly where it binds. So we can use this screening so that we can have an idea where it actually binds. Another one is the orphaning a receptor. We found, or usually done when we discover new receptors, but we do not really know what it is for. So we can try to dock several known compounds here so that we have an idea what is that for. So another one is for protein-protein interactions. So the two different proteins, how do they interact with one another? And some structure function studies. Because in biochemistry and in biology, molecular biology itself, the tenet is once the structure of the molecule is the reason why it's functioning. So it gives us a more insight because this molecular docking is more of a functional type of discovery. So it gives us an insight whether how this structure affects the function of this activity. It can also be used for some enzymatic reaction mechanism. So for example, if we dock this one on this molecule, just like a receptor with a target, so we can we inhibit the activity of that enzyme. And also in protein engineering, if you want to design and engineer novel proteins for specific functions, we can use this docking. So what is the basic principle of docking? So here we basically have two important components, parts, how can I say, requirements for the docking. So first we need to have a target. The target is your receptor molecule. As I say here, the receptor molecule is very large. So an example is the protein. So here is the structure of your protein. So it can be an enzyme, it can be a channel, an ion channel, for instance, a receptor on the surface of the cell, and your ligand is very small compared to that. This is your ligand. So if we look at our ligand in contrast to the receptor, actually this is for usual receptors or for usual protein molecules, these molecules actually on the smaller side. You will notice that there are very, very large molecule and we have very, very small ligands. So these ligands actually quite large. So if you notice, we have a large molecule and a smaller ligand. That is why it's important to identify the receptor and the ligand. So we have this target and this is ligand. We dock them. That means we want to know how this ligand would fit into your target. So you have this, the region where it fits is the binding site. So knowing the binding site is the third and another important requirement for docking, because the binding site is a specific and unique site. The ligand does not really, in reality, the ligand does not bind just anywhere. It doesn't bind here or here or here. It has a specific binding site. So that is why in doing docking studies, we must be first be able to identify or at least have an idea where the binding site is. If we really have no idea, we can have several candidate binding sites, try to dock the molecule there, and which binding site gives the best fit for that molecule. So it can be done like that. If it's a blind docking procedure. But usually we do have studies beforehand where we can identify the docking site. The binding site. Now, after the docking, this is all done virtually by the computers. So there is a calculation of the binding energy. This is basically a score. This is not the absolute value, but usually for most docking software, it gives us a value of quote unquote binding energy. Other softwares just give you a score. A score is based on how that particular docking software is written, based on the calculation used by that particular docking software. So each binding score is unique to that software and even to that particular run. So that means, for example, you run your compound today, and then you run it again tomorrow on the same, exactly the same activity. Sometimes your score is not exactly the same. It might be near, but not always exactly the same. It depends on the calculation of the structure. But the score gives us an idea how tightly uh the binding, how close is the affinity or how strong is the affinity of the target to the ligand. So usually some software, if it's an in-house score, a high score means strong binding, so it binds tightly. A low score is weak binding. But sometimes some softwares give you, instead of a score, it's a binding energy. If the binding energy is the one that is given, usually it's in the value of kilocalories or kilojoules per mole. This doesn't mean that it's the actual energy of binding, the energy that binds this ligand to the target, but it gives you just an idea of the affinity. And if it's given as a binding energy, usually the lower the binding energy, the more negative the binding energy, the more tightly the ligand binds to the receptor based on the prediction of the docking software. Keep in mind, molecular docking is mostly predictive. So you are predicting whether a ligand will bind to your target. Okay, so the general method is usually we get the structure of your receptor. It's important that it should be the structure. So we can get it through a database. Usually it's the RCSB database, or we can predict even the structure. But it's just that we really need the structure. Now, this is your receptor. This is your protein. We call it the receptor. We call it the protein. We call it the target. It all means the same. And then we have the ligand. The ligand is a small compound or a smaller molecule. It can be a peptide. It can even be a drug. It's a small molecule. This is, if you have chemistry, these are structures of your organic compounds. These are the structure of your drugs, molecular structure of the drugs. And then you perform the docking. So you specify a specific binding site, not you do not just input the ligand, the receptor and the ligand, but you also need to define a particular site where your ligand would bind. Okay, and this is the general method for docking. Now, what does the docking software do? The docking software is basically searching for the best pose, just like how this model here is posing in the camera, many different poses, and the final evaluation is finding the best shot or the best pose. Your computer is moving your ligand around. So it's usually the ligand that is moving around. That is why it's important when you input the data, you input the ligand on the correct box and the receptor on the correct box. You do not switch them around, because if you switch them around, it would be difficult. Uh, it's because the receptor usually is stationary while your ligand, the computer software for the docking moves your ligand around in different poses. So until it finds the best fit, or the best, basically the best shot or the best fit for your particular binding site. So another example here. So here you have several, this GIF image is doing several poses until it finds the best fit pose for your binding site. Now this is the binding site. This is the gray ones. This is your receptor. This is, if you notice, it's the receptor here is on the pink and blue, and then this one is in the gray, and it's represented differently. The mostly this is just based on the visualization software, like Chimera. You can input it whether you want it in the ball and stick figure or somewhat like this, just surface, showing the surface only of the molecule. But all in all, in the docking software, regardless of what representation it uses, it's the calculation stays the same. It's just that your software is moving your ligand around until it finds the best fit. So in this laboratory activity, we will do some applications of your ligand. So our receptor, our protein, our target is actually a voltage gated sodium channel. So NaV. So that means it opens when a certain membrane voltage, basically when the charge of the membrane becomes positive, or at least it reaches the threshold, which is -50. So it reaches the -50 threshold, it opens, and it allows, as a sodium channel, it allows the passage of the sodium ion, so it opens. So this is your voltage gated sodium channels. So this is our structure, or rather our target for this laboratory activity. So, and we will be looking at several inhibitors of our voltage gated sodium channels. Now, here are some inhibitors: guanidinium toxins, peptide toxins, the pore blockers. We have this VSD4 inhibitor. So here, A, B, C, and D, each of these inhibitors binds to a specific binding site. It doesn't just bind anywhere. They do have a specific binding site. So for example, the guanidinium toxins binds around in this area, the peptide toxin is in this area, and the pore blockers in this area, the VSD4 inhibitors in this area. But all in all, they have similar functions. They can inhibit the activity of the channel. It can inhibit the opening of the channel, or it can inhibit the closing of the channel. Uh, depending on its binding and its activity. Why does, for example, how does it happens that even if they bind to different sites, they can have the same effect? It depends on when they bind to that particular molecule, or your target, it induces a change in the structure of your target. And that induced structural change causes your target to become permanently open or permanently closed, depending on what that compound is for. And of course, the structural change on your compound. Okay, so in this laboratory, we will be looking at several anesthetics. The first one is lidocaine. You have already used that in your frag sciatic nerve studies, but we also look at several other anesthetics and neuroactive drugs. In fact, these four other drugs are structurally similar to lidocaine. We will dock all five of them, and we will have lidocaine as our control. That means lidocaine, we already know that this is a pore blocker. We will this is our basically a control. It's important in dockings because, as I said, the values or the scores is given is based on the calculations of the software. So we need to have a proper baseline, and lidocaine as our control will give us a baseline of the docking. So we can, using our control as lidocaine, we can compare how strong the affinity is for prilocaine, etidocaine, tocanide, and nefiracetam. Okay, so these are anesthetics commercially. Not all of them are anesthetics. One of them is a drug for Alzheimer's, but they have structural similarity to lidocaine. So in this laboratory, you can evaluate what first of all, look at the commercial application of each drug and which is the strongest binding and the least binding. Okay, so I'm not saying that all of them actually is used for voltage-gated sodium channel, but because they do have some structural similarity, so they might have some affinity. We will discover that in this laboratory. Okay, and in the next part, I will show you how to do docking in with using Chimera and our software that we will be using, which is the docking incrementally, or DINC software.